Hello there. Do you know about DNA? It's actually pretty fascinating stuff. DNA is short for deoxyribonucleic acid, and it holds all the genetic information necessary for living organisms, including animals, plants, and even viruses. DNA is made up of two strands of nucleotides that are held together by covalent bonds. Each nucleotide has a nitrogen-containing base, a sugar called deoxyribose, and a phosphate group. A and T pair up while C and G pair up, forming a double helix structure that looks like a ladder. The order of these base pairs contains the genetic information, and both strands of DNA have the same information. When the strands separate, the genetic information gets replicated. However, it's interesting to note that most of the DNA in our cells doesn't actually have a function. In eukaryotic cells, DNA is arranged into structures called chromosomes. Before a cell divides, its chromosomes are duplicated through DNA replication, ensuring that each new cell has a full set of chromosomes. Eukaryotes store their DNA in the nucleus or in mitochondria and chloroplasts, while prokaryotes store it in the cytoplasm. When it comes to making proteins, DNA is first transcribed into RNA through a process called transcription. RNA has the same nucleotide bases as DNA, except it has uracil instead of thymine. This RNA is then translated into proteins, based on the genetic code. Chromatin proteins, such as histones, help compact and organize DNA within eukaryotic chromosomes, which determines which parts of the DNA are transcribed. Hey there! Let me give you some cool facts about DNA. DNA is a long molecule that's made up of nucleotides, which consist of a backbone and a nucleobase. It has a double helix structure, which means two helical chains held together by hydrogen bonds. Each nucleotide is composed of a nucleobase, a sugar, and one or more phosphate groups. The backbone of a DNA strand is made up of alternating phosphate and sugar groups. Fun fact: DNA sugar is called 2-deoxyribose, while RNA sugar is ribose. In DNA, there are 4 nucleobases: adenine, cytosine, guanine, and thymine. These nucleobases pair up to form a complete nucleotide, and they are classified into 2 types, purines and pyrimidines. In RNA, thymine is replaced by uracil. DNA can be single-stranded or double-stranded, and the stability of dsDNA depends on factors such as GC content, sequence, and length. DNA can also be twisted like a rope, which is called DNA supercoiling. In a DNA double helix, complementary base pairing occurs, with adenine bonding only to thymine in 2 hydrogen bonds, and cytosine bonding only to guanine in 3 hydrogen bonds. The direction of the twist in the helix can result in positive or negative supercoiling. There are different conformations that DNA can take, such as aDNA, bDNA, and zDNA forms. Quadruplex structures are formed at the ends of the linear DNA molecule. Lastly, sense and antisense sequences can exist on different parts of the same DNA strand. The conformation that DNA adopts depends on several factors such as hydration level, DNA sequence, amount and direction of supercoiling, chemical modifications of the bases, and the presence of polyamines in solution. Hey there! Let's talk about chromatin, the nifty little structure that wraps our DNA into chromosomes. The way DNA is organized within chromatin can impact how our genes are expressed. Interesting, right? One factor that can affect packaging is base modifications. Regions of chromatin with low or no gene expression often contain high levels of cytosine base methylation. Additionally, modifications to the histone proteins around which DNA is wrapped can influence gene expression too. Now, on to mutagens. These sneaky little things can modify the DNA sequence in all sorts of ways, depending on the type of mutagen. For example, UV light can cause thymine dimers, while oxidants like free radicals or hydrogen peroxide can create multiple forms of damage, including base modifications and double-strand breaks. Some mutagens like to hang out between adjacent base pairs in a process called intercalation. Ethidium bromide, acridines, and doxorubicin are all examples of these sneaky mutagens. But in order to fit between base pairs, the bases need to separate, which unwinds the double helix and distorts the DNA strands. Not good. This can mess with both transcription and DNA replication, leading to mutations and toxicity. Because of this, DNA intercalators can be carcinogens. There. Let's chat about DNA and how it works. Eukaryotes, which include humans, typically have DNA that exists as linear chromosomes, while prokaryotes usually have circular chromosomes. All of the chromosomes in a cell make up its genome, and in humans, we have 46 of them. The genetic information is held within genes, which are little fragments of DNA. During transcription, a cell duplicates the information in a gene by copying the DNA sequence into a complementary RNA sequence. This RNA copy is then used to create a corresponding protein sequence during translation. DNA replication, the process in which a cell copies its genetic information, is possible because of the double-stranded structure of DNA. To fit into the small available cell volumes, genomic DNA is tightly and orderly packaged through DNA condensation. In eukaryotes, DNA is located in the cell nucleus, with smaller amounts in mitochondria and chloroplasts. In prokaryotes, DNA is held within an irregularly shaped body in the cytoplasm called the nucleoid. Genes, which control particular characteristics in an organism, include an open reading frame that can be transcribed in regulatory sequences, such as promoters and enhancers, which regulate transcription. The sequence of bases in a DNA strand within a gene determines a messenger RNA sequence, which subsequently identifies one or more protein sequences. The genetic code, made up of three-letter codons created from a sequence of 3 nucleotides, establishes the relationship between nucleotide sequences and amino acid sequences. During transcription, RNA polymerase copies the codons of a gene into messenger RNA. The ribosome then decodes this RNA sequence by base pairing the messenger RNA to transfer RNA, which carries amino acids. There are 64 possible codons made up of 4 bases in 3-letter groupings, with the majority of amino acids having multiple possible codons. There are also 3 stop or nonsense codons that signify the end of the coding region. For an organism to grow, cell division is necessary. When a cell divides, it must replicate the DNA in its genome so that the 2 daughter cells possess the same genetic information as their parent. DNA replication is straightforward because of the double-stranded structure of DNA. The 2 strands are separated, and each complementary DNA sequence is produced by an enzyme known as DNA polymerase. This enzyme locates the correct base through complementary base pairing and attaches it to the original strand to make the complementary strand. Different mechanisms are used to copy the antiparallel strands of the double helix, and the result is a perfect replica of the DNA. Hey there! Let me explain to you in a more friendly and conversational way about how proteins interact with DNA. So, there are different types of proteins that bind to DNA and have different roles. Firstly, there are some proteins that don't specifically interact with DNA but are responsible for organizing it into compact structures called chromatin. In eukaryotes, this role is played by histones, while prokaryotes involve multiple types of proteins. High mobility group proteins also bind to chromatin and help to bend arrays of nucleosomes and arrange them into larger chromosome structures. On the other hand, some proteins are specifically designed to bind to single-stranded DNA. These proteins, like replication protein A in humans, help in the processes of DNA replication, recombination, and repair. They stabilize single-stranded DNA and prevent it from forming stem loops or being degraded. Lastly, some proteins are evolved to bind to specific DNA sequences. These are called transcription factors and are responsible for regulating gene expression. Each transcription factor binds to a particular set of DNA sequences and activates or inhibits gene transcription. They can do this by either binding directly to the RNA polymerase or by binding to enzymes that modify histones at the promoter, making the DNA template more or less accessible to the polymerase. I hope that made it easier for you to understand. Hey there! Let's talk about some of the enzymes involved in DNA processes, but in a more friendly and casual tone. Firstly, we have nucleases. These enzymes help cut DNA strands by catalyzing the hydrolysis of phosphodiester bonds. There are two types of nucleases: exonucleases, which remove nucleotides from the ends of strands, and endonucleases, which cut within the strands. Some endonucleases, like EcoRV, are used in molecular biology because they can cut DNA at specific sequences. They are also part of the bacterial restriction modification system, which helps protect against phage infections. Scientists also use them in DNA fingerprinting and molecular cloning. Another enzyme that's important is DNA ligase. This enzyme can rejoin broken DNA strands, which is critical for DNA replication, repair, and genetic recombination. In lagging strand DNA replication, DNA ligase joins short DNA segments to create a complete copy of the DNA template. Topoisomerases are enzymes that have both nuclease and ligase activity. They regulate the amount of supercoiling in DNA, and some of them work by cutting the helix and allowing one section to rotate, reducing its level of supercoiling. Others can cut one helix and pass a second strand through the break before rejoining the helix. These enzymes are essential for DNA replication and transcription. Helicases are molecular motor proteins that unwind the DNA double helix into single strands by breaking hydrogen bonds between bases. They use chemical energy in nucleoside triphosphates, mainly ATP, to do this. Helicases are essential for many processes where enzymes need access to the DNA bases. Polymerases are another essential group of enzymes that synthesize polynucleotide chains from nucleoside triphosphates, with the sequence based on existing templates. They add nucleotides to the 3' hydroxyl group at the end of the growing polynucleotide chain and work in a 5-3 direction. Polymerases accurately synthesize the complementary strand of their template and are classified by the type of template they use. DNA-dependent DNA polymerases copy DNA polynucleotide chains in DNA replication. They have proofreading activity, which corrects occasional mistakes in synthesis by removing incorrect bases. RNA-dependent DNA polymerases copy RNA strands into DNA, such as reverse transcriptase, which is a viral enzyme involved in retrovirus infections, and telomerase, which is essential for telomere replication. Finally, transcription is carried out by a DNA-dependent RNA polymerase, which copies DNA sequences into RNA. It binds to a DNA sequence called a promoter to start transcribing a gene and separates the DNA strands. The RNA polymerase copies the gene sequence into an mRNA transcript until it reaches the terminator region, where it halts and detaches from the DNA. RNA polymerase II is responsible for transcribing most of the genes in the human genome and operates as part of a protein complex with regulatory and accessory subunits. Hey there! Let me break down the science of DNA recombination for you in a more pleasant way. So, in human cells, DNA helices tend to stay apart from other sections of DNA, each chromosome occupying its own territory in the nucleus. This is important for the safe storage of genetic information. However, during sexual reproduction, something called chromosomal crossover happens, which is also known as genetic recombination. This is when two DNA helices break, swap and rejoin, exchanging genetic material between chromosomes. This process has a lot of benefits, including creating new combinations of genes, which can drive the evolution of new proteins and promote natural selection. Genetic recombination can also help with DNA repair, especially when cells experience double-strand breaks. There are enzymes called recombinases, like RAD51, which help with the recombination reaction. The most common type of chromosomal crossover is called homologous recombination, which happens when two chromosomes with very similar sequences exchange genetic material. However, non-homologous recombination can be harmful, leading to chromosomal translocations and genetic abnormalities. The recombination process itself involves several steps. It starts with a double-stranded break, caused by an endonuclease or DNA damage. Then, the recombinase catalyzes the formation of at least one holiday junction, which is a tetrahedral structure that moves along the chromosomes, exchanging one strand for another. The process ends with the cleavage of the junction and religation of the DNA. It's important to note that only strands with the same polarity exchange genetic material during recombination. There are two types of cleavage: north-south cleavage, which nicks both strands of DNA, and east-west cleavage, which leaves one strand intact. Overall, the formation of a holiday junction during recombination enables genetic diversity, the exchange of genes on chromosomes, and the expression of wild-type viral genomes. Hey there! Did you know that DNA holds all the information necessary for life to function, grow, and reproduce? It's pretty amazing. However, scientists are still unsure how long DNA has been the primary genetic material during the 4 billion-year history of life. Some believe that RNA may have been the original genetic material, with RNA playing a crucial role in early cell metabolism due to its ability to transmit genetic information and carry out catalysis as part of ribozymes. This RNA world may have influenced the current genetic code we see today. Interestingly, the number of nucleotide bases in early organisms was likely a trade-off between replication accuracy and ribozyme catalytic efficiency. While a small number of bases may have increased replication accuracy, a larger number of bases may have improved ribozyme catalytic efficiency. However, there is no direct evidence of ancient genetic systems because DNA is not recoverable from most fossils. The building blocks of DNA, such as adenine and guanine, may have even formed in outer space. Organic compounds like uracil, cytosine, and thymine, which are found in DNA and RNA, have also been formed in laboratories under conditions similar to those found in outer space. These conditions include starting chemicals like pyrimidine, which has been found in meteorites. Pyrimidine and other carbon-rich chemicals, like polycyclic aromatic hydrocarbons, may have formed in red giants or interstellar cosmic dust and gas clouds. Scientists reported that they had sequenced DNA from animal remains for the first time. The oldest DNA ever sequenced came from a mammoth that was over 1 million years old. Isn't that incredible? Bioinformatics is an exciting field that involves creating techniques for organizing, manipulating, and analyzing biological data, including DNA sequences. These techniques have led to remarkable advancements in computer science, particularly in string searching algorithms, machine learning, and database theory. By using string searching algorithms, we can locate specific nucleotide sequences within larger ones, and we can compare DNA sequences with others to identify similar sequences and unique mutations. These methods help us study the evolutionary relationships among species and how proteins work. Gene-finding algorithms are another tool used in bioinformatics that can identify regions of DNA that code for proteins or RNA molecules. This allows us to predict which genes are present in an organism before performing experiments. Additionally, comparing entire genomes can provide valuable insights into an organism's evolutionary history and help us study complex evolutionary events. DNA nanotechnology is an exciting field that harnesses the unique properties of DNA and nucleic acids to create self-assembling branched DNA complexes with useful properties. We can also use DNA structures to arrange other molecules, like gold nanoparticles and proteins, and use synthetic oligonucleotides, known as aptamers, to target specific molecules in biotechnology and biomedical applications. Phylogenetics is a powerful tool in evolutionary biology, allowing geneticists to infer the evolutionary history of organisms by comparing DNA sequences. Population geneticists can also study the history of particular populations by analyzing DNA sequences, which is useful in fields ranging from ecology to anthropology. Finally, DNA has great potential as a storage device because of its high storage density. However, practical use has been limited due to high costs, slow read and write times, and insufficient reliability. Nonetheless, ongoing research aims to overcome these limitations and unlock the full potential of DNA as a storage medium. Hey there! Let's talk about the different methods scientists use to work with DNA. They've come up with some pretty cool techniques to purify DNA from different organisms. One way they do this is called phenylchloroform extraction. They also use methods like restriction digests and the polymerase chain reaction to manipulate DNA in the lab. Scientists can even create new DNA sequences by putting together different DNA sequences, which they call recombinant DNA. They can then introduce these artificially created sequences into organisms using plasmids or viral vectors. This leads to the creation of genetically modified organisms, which can be used to make proteins for medical research or grown in agriculture. In forensic science, DNA found at a crime scene, such as in blood, semen, skin, saliva, or hair, can be used to identify a suspect. This is done through a process called DNA profiling or DNA fingerprinting. Forensic scientists compare the lengths of different sections of repetitive DNA, like short tandem repeats and mini-satellites, to find a matching DNA profile. This is a very reliable technique for identifying a suspect. There are also things called deoxyribonucleics, which are single-stranded DNA sequences that scientists can isolate from a large pool of random DNA sequences using a technique called in vitro selection or helix. These DNazymes are pretty cool because they can catalyze a variety of chemical reactions, like RNA-DNA cleavage, RNA-DNA ligation, amino acid phosphorylation dephosphorylation, and carbon-carbon bond formation.